In this video, we are going to learn about some unrelated topics together in molecular biology. So let's start. Prokaryotic genome, Escherichia coli, basic features. Chromosome, plasmid, nucleotide. So introduction, the term prokaryote means primitive nucleus. Cell in prokaryotes have no nucleus. The prokaryotic chromosome is dispersed within the cell and is not enclosed by a separate membrane. Much of the information about the structure of DNA has come from the studies of prokaryotes because they are less complex genetically and biochemically than eukaryotes. Prokaryotes are monoploid, they have only one set of genes and copy of genome. In most viruses and prokaryotes, a single set of gene is stored in a single chromosome, single molecule, either RNA or DNA. Prokaryotic genomes are exemplified by the E. coli chromosome. The bulk of the DNA in E. coli cells consists of single closed circular DNA molecule in length 4.6 million base pairs. The DNA is packaged into a region of cell known as the nucleoid. Prokaryotic cell, ribosomes, mesosomes, plasma membrane, cell wall, capsule, plasmid, bacterial flagellum, pili, then cytoplasm, nucleoid. Components of prokaryotic cell: plasma membrane, an outer covering that separates the cell's interior from its surrounding environment. Cytoplasm, a jelly-like cytosol within the cell in which other cellular components are present. DNA, the genetic material of the cell. Ribosome, where protein synthesis occurs. Genome organization in prokaryotes do not contain nucleus and membrane-bound organelles. Simple genome organization: genes usually do not have introns and histone particles. Circular double-stranded DNA molecule, but may be a linear molecule with some major groups. Supercoily. Then viruses, capsid viral genome, prokaryotes, plasmids, bacterial chromosomes. Introduction to genetic organization. In the cell, DNA is associated with proteins, and each DNA molecule and its associated protein is called a chromosome. This organization holds true for prokaryotes and eukaryotic cells. Cell DNA, nucleus, chromosome, like this. Then packing of the DNA into chromosomes. The chromosome is a compact form of DNA that readily fits inside the cell. It serves to protect the DNA from damage. Only DNA packaged into a chromosome can be transmitted efficiently to both daughter cells when a cell divides. Genetic organization of prokaryotic. Prokaryotic cells have a single chromosome and it is usually present in the cytoplasm. Now, prokaryotic cells having multiple chromosomes are identified. Packing of DNA. Prokaryotic cells typically have smaller genomes. The need to compact their DNA is still substantial. E. coli must pack its 1 mm chromosome into a cell that is only 1 meter in length. It is less clear how prokaryotic DNA is compacted. Proteins associated to DNA, bacteria have no histone or nucleosome. If they do have other basic proteins that they serve similar functions, then multiplication of prokaryotic DNA, when prokaryotic cells are dividing rapidly. However, portions of the microchromosome in the process of replication are present in 2 and sometimes even 4 copies. Genome size. Prokaryotic versus eukaryotic genome. Prokaryotic genome. They are small in size, contain single circular chromosome. They are haploid. DNA is not associated with histones. They are condensed in nucleoid via DNA supercoiling. Eukaryotic genome. They are larger than prokaryotic genome. Contain multiple linear chromosomes. They are diploid. DNA is associated with histones. They are condensed in membrane-bound nucleus via histones. E. coli, 89% coding, 4289 genes, 122 structural RNA genes, operone polycystonic transcription units, haploid circular genome, usually asexual reproduction, transcription and translation take place in the same compartment. Basic feature of E. coli, named for Theodor Ehrlich, German physician. They are short-ruled. They are facultative anaerobes. They can ferment lactose. Germ-negative type of classification. They are also motile. They are having capsule layer. We can culture them. Makunki agar. I'm not sure about the pronunciation. They are pathogenic or non-pathogenic. Many strains are there. O antigen, somatic 171 antigens, H antigen, flagella 56 antigens, K antigen, capsular, fimbriar antigen, 80 antigens, like this. Diseases caused by E. coli. E. coli is responsible for 3 types of infections in humans: urinary tract infections or UTI, neonatal meningitis, intestinal diseases like gastroenteritis. Next topic, replication of chromosome terminal in eukaryotes. Introduction. Telomeres. Telomeres are regions of DNA at the end of linear eukaryotic chromosomes required for the replication and stability of chromosomes. Thus, a repetitive nucleotide sequence at each end of eukaryotic chromosome protects the chromosome from degradation. These are placed in such a way that it appears like specialized DNA caps that prevents the genes at chromosome ends from getting deleted as cells continue to divide. Telomeres consist of hundreds or thousands of repeats of some short DNA sequences, which varies between humans and other mammals. Cell, nucleus, chromosome, telomere, DNA, you could see. Telomerase enzyme. Some cells have the ability to reverse the telomere shortening. This is done by expressing an enzyme called telomerase enzyme. Telomerase extends telomerase to chromosomes. Telomerase is an RNA-dependent DNA polymerase, hence it can make DNA using RNA as a template. Telomerase is important for maintaining chromosome integrity. The ends of chromosomes are also maintained by the action of telomerase enzyme. Telomerase enzyme is a ribonucleoprotein enzyme. 2 essential core subunits are there, like telomerase RNA, TER, telomerase reverse transcriptase protein, TERT. TER and TERT. TERT resembles with TER to repeatedly synthesize tandem repeats of DNA of telomeric end of the chromosome. Replication of telomerase in eukaryotes. DNA polymerase cannot replicate and repair DNA at the end of linear chromosome. The ends of linear chromosome are called telomeres, replicate by the expression of an enzyme telomerase. Telomerase attaches to the end of chromosome. Complementary bases of RNA template are added on the 3' end of DNA strand. Lagging strand gets elongated by telomerase. DNA polymerase add complementary nucleotides or RNA primer to end of the chromosome in presence of primase, resulting in replication of telomerase. The telomerase enzyme attaches at the end of chromosome telomere, which contain a catalytic part and built-in RNA template. Whereas the enzyme binds to a special RNA molecule that contains a sequence complementary to the telomerase repeat. The telomerase enzyme extends by adding nucleotides to the leading strand, the telomerase RNA, using complementary RNA as a template. When the lagging strand is long enough, a matching strand or a complementary strand can be made by normal RNA replication. The normal RNA replication is processed by RNA primer in presence of enzyme RNA, DNA polymerase and primase, thus producing a double-stranded telomeric DNA. Telomerase have repeated sequences that are complementary to the internal RNA template or the telomerase enzyme. Therefore, the internal RNA template base pairs with the very tip of the single-stranded portion of the telomere. The reverse transcriptase activity of telomerase uses the internal RNA as a template for the lengthening the single strand of the telomere. When the single strand of the telomere is long enough, primase can synthesize a primer and DNA polymerase can extend the primer with DNA. So conclusion, telomerase is not usually active in most somatic cells of the body, but it is active in germ cells, cells that make sperm and egg, and some adult stem cells. These are cell types that need to undergo many divisions, or in the case of germ cells, they give rise to new organism with a telomeric clock. Many cancer cells have shortened telomeres, and telomerase is active in these cells. If telomerase could be inhibited by drugs, the part of cancer therapy, their excess division and thus the growth of the cancerous tumor can potentially be stopped. Cells that undergo cell division continue to have their telomerase shortened because most somatic cells do not make telomerase. Telomerase shortening is associated with aging. Telomerase reactivation in telomerase deficient mice causes extension of telomeres. This may have potential for treating age-related diseases in humans.